And now we move on with the next speaker. And the next speaker is Dr. Natarajan. He will give a talk on exome-sequencing of vitiligo lesions indicate lower burden of somatic variation, implication in risk for non-melanoma skin cancer. Please.
Very good morning, and I really look forward to the discussions that we had, and maybe towards the end of the talk I will continue to show you some data and the discussions can continue. And to just give an introduction about myself and what our lab does. I mean, we are essentially a group of researchers. We are in the north of Delhi. I mean, we are in the northern part of India. And if you end up coming to Delhi, please do visit our lab. I mean, we work on two different aspects, primarily concerning vitiligo. I mean, we take a top-down approach in understanding what are the changes that have happened in the vitiligo skin, the depigmented skin, to try and understand, the pathophysiology of vitiligo. Over the years, as a larger team, we've had multiple insights. I know many of them have been discussed during the last 2 days, so I'll not go into the details about, how we have contributed in that regard. But more about, continuing the conversation that just preceded my talk was about how do we really go back to melanocytes and alter these cells to really cause the repigmentation. And towards this, the approach that we all in the lab have been taking is actually to understand the continuum of changes that happens in these cells. While we think of melanocytes are pigment-producing factories, they are in fact known to be present in different forms that we know of. We know of melanocytes as migratory, melanocytes as a melanoblastic stage, a melanocyte that can respond to UV and make pigment in a very short span of time. These are all different states is what we believe, and therefore identifying the population that's responsible for repigmenting in the interfollicular epidermis would come a long way in causing the repigmentation. So therefore, while the vitiligo-based evaluation of depigmented and the lesional and non-lesional skin is more of a top-down approach, we try and take a bottom-up approach with respect to understanding the cell fate transitions in melanocytes, and we hope that in some time, maybe in a couple of years, we'll be able to really merge the two at some point in time and be able to provide a more actionable therapeutic intervention in vitiligo. So with that, we've all spoken about how vitiligo, there are multiple kinds or phases of the disease, and maybe no two vitiligo or no two lesions on the same patient may in fact be very identical in terms of the response to treatments, etc. So it appears that there is an evolution of the disease, and we need to understand what are the ways in which the disease progresses. So while that happens to be the major area of interest, I'll just switch to what I just decided to talk about in today's talk. That's on a completely different aspect, which is very relevant for the disease, but nonetheless has remained controversial. So please feel free to dissuade me from really going ahead on this, because there are a lot of things that have not remained very explainable when it comes to the pathology of vitiligo. So one of the clinical observation that has come repeatedly is the decreased risk of non-melanoma skin cancers in subjects with vitiligo. And this is rather counterintuitive because patients with albinism have a very high risk of getting non-melanoma cancers, and therefore it is logical only to believe that people with vitiligo would also have an elevated risk. And multiple studies, when systematically have studied the incidence of non-melanoma cancers, these incidents have found to be either comparable or in many cases found out to be lower than subjects who don't have vitiligo. So while this has been mostly a demographic-based study, one needed an observation at a molecular level to be able to understand whether it is indeed true. If so, what would really mean? So therefore I really want to divide the talk into two parts to really say, can we really identify molecular correlates of substantiation of decreased risk of non-melanoma cancer in subjects with vitiligo? Towards this, something that caught our attention was a paper that published in 2015, and that's a rather interesting study where people have studied the skin punch by biopsies and isolated the genomic DNA and looked at the cancer-associated genes in much deeper sequencing-based method and identified the minor allele changes in somatic variations in these skin punches. And because skin happens to be a two-dimensional structure and the clonal populations of variations that arise spontaneously in the skin are likely to be present locally, this offers a very nice way, the genomics offers a nice way to really look at these somatic variations and estimate the sizes of mutant clones that arise in the normal skin. And what came out as a rather surprising observation, this was subjects, healthy subjects, seemingly normal individuals. They also had harbored several mutations which can be potentially cancer-causing from a normal, healthy individual, normal pigmented skin. So if this was the case, can we apply this kind of an approach to a vitiligo lesional skin to get insights about what's going on with the vitiligo depigmented lesions? To be able to do that, I mean, this is what is a schematic of, with the logic behind what we do. So if there are clones that arise because of somatic variations, these clones that arise because of variations, they would expand because of selection, and with time, you would be able to identify the size of the clones by estimating the somatic variation frequency in these cancer-causing genes. So these were the questions that we asked. How do lesional vitiligo skin compare with the matched non-lesional skin for the somatic variations? And while these changes are there in the skin, can we identify potentially the reasons why there could be these kind of observations that can tell us about the alterations in the somatic variation frequency? This was a study design. We had taken the blood, exposed lesion, and the unexposed non-lesion skin from the same individuals. We chose individuals who had lesions in the extremities of the body, and also the non-lesional skin was from the gluteal area, which is relatively unexposed. The blood was used as a baseline to look for variations that are coming from the blood, and therefore any variations from the blood were removed. And now we had, from these 6 individuals, localized somatic variations in either the non-lesional or the lesional skin, and we concentrated on cancer-associated genes because that's what is the meaningful thing to do in this case. So this is really the methodology for that. The exome sequencing was done in something like 450 to 500x to be able to capture smaller clonal populations that arise spontaneously. So this is essentially how we compare our data with what's there in literature. The spread of somatic variation burden in multiple cancers is tremendous. And what's seen here is the healthy skin data that's there, that was published in 2015, that is presented. And these are the two, the non-lesion and the lesional skin from the vitiligo subjects. The spread appears comparable, but when we look at the pairwise comparison between the somatic mutation burden, which is shown here on the right, you would see that in pretty much 5 out of 6 individuals, the non-lesional skin has a higher somatic mutation burden in cancer-associated genes. And barring one individual, there were more somatic variations in the non-lesional skin, which is indeed a pigmented, unexposed skin. And that really sort of was statistically significant and gave us the confidence that perhaps the demography-based observations that people had studied, there is indeed a molecular correlate to that. And this is essentially a different representation of the data. So where we are looking at the different genes and somatic mutation burden in these cancer genes, not just the number of clonal populations, but also the overall genes in which these variations arise in vitiligo subjects was much lower. Why could this be arising is something that we went on to check. And for that, we looked at the transcriptomic profiles. And we looked at, again, the non-lesional and lesional and non-lesional RNA. And one of the processes that we expect to be decreased, for example, decrease in the pigmentation or melanocytic transcripts, and that was really observed. But what's projected here is the upregulated set of processes. And the ones that are really caught our attention are the proliferation or the nuclear division and the response to DNA. These two processes are heavily enriched in the lesional skin. So are there any evidences for this at the cellular level? Indeed the case. I mean, when we look at some of the proliferation markers, we do find them higher in the lesional skin. And many of the DNA cell proliferation genes are elevated in vitiligo lesions. And also, many of the DNA repair genes were upregulated. What's there on the right side is in fact called a volcano plot, and most of the genes pertaining to DNA repair, something like 34 out of 37 were upregulated. To look at if there are any cellular changes associated with DNA repair, we performed COMET analysis, which are essentially nicks, estimating nicks in the DNA, and the nicks in the DNA is more in the non-lesional skin than the lesional skin. So all these pointed out to the fact that perhaps the demography-based observations are in fact something that we need to really look at them at the molecular level more closely. And what also caught our attention was that in cancer field and also in the cultured keratinocytes, it's now becoming evident that there are different kind of proliferative populations, and these proliferative populations have been called as meroclones, holoclones, or paraclones. And one of the hallmark features of these holoclone populations is their ability to repair DNA with high proliferative capacity. So when we map the transcriptional signature of vitiligo lesions, we find that in fact the holoclone populations are expanded in vitiligo lesions. So therefore, with that, what we really come to the conclusion is that there are changes. The somatic variation burden in cancer genes are in fact low, and that's a good news for subjects with vitiligo, because one of the long-term effects of exposure to UV is cancer progression, and perhaps we don't need to worry so much about it. But does that mean that we don't have to worry about photoprotection at all? Of course, that's not what is being projected. That's not what we intend to say, because there are other effects, harmful effects of photo damage that needs to be taken care of. And also, does it really mean that this is the pilot study based on the Indian population? Does it really hold true with other populations? Remains to be seen. And how does it compare with subjects who do not have vitiligo? Those are something that it needs to be done. And perhaps when we do that, we'll be able to say this with a lot more confidence, and we'll be able to take the study ahead. With that, I'll just come back to the conversations that we had in the previous discussions. There are models that can study immune progression. Mice happens to be a beautiful model. Professor Harris showed us how he has been extensively using these studies. But for repigmentation, there has been a long loss. There is a much needed demand for an animal study that can tell us about spontaneous repigmentation. So some of the work that
we have to reach the conclusion, because we are out of the time.
I'm done. So this is essentially a work where we have looked at the spontaneously repigmentation model in buffalo as a good model for vitiligo. Many of the clinical features seen in subjects are recapitulated in this. And also we study zebrafish in the model, where we can look at melanocyte-specific changes. With that, I'll thank the organizers and the chair for taking me a couple of extra minutes. Thank you so much.
Thank you very much. Any question or comments?